chapter 11 and 12, we are starting the nervous system. The nervous system can be divided into two parts. Central nervous system and peripheral nervous system. Central nervous system is going to be everything in the middle. Peripheral is going to be everything on the outside. Okay, like your limbs and so on. So chapter 11 strictly focuses on the peripheral nervous system. Okay, there's going to be a short intro on it where it tells you that overall what does the nervous system do? It communicates. It's responsible for controlling. The very first lecture we had together, I asked you which system was most important, and typically most people say nervous or cardiovascular. And I told you that they're all equally important. I think somebody in here said that. We had this conversation already. But the nervous system, it communicates, and it communicates using electricity. So the communication the signals that it sends out go out very quickly, travel fast, but they dissipate very quickly as well. Okay, do you remember the other system that communicates? Endocrine, uses hormones, but that's an AMP too. Okay, so it talks about how it uses electricity in order to communicate. Here it talks about the difference between sensory and motor input/output. I mentioned this in a previous lecture, but here it is one more time because you're going to see this pattern. And I'm going to point out things as we get started in the nervous system. Sensory is always going to travel up because that information always, when you sense something, it has to travel towards your central nervous system. So we say it's afferent. Motor is a message you telling your body to do something, so it travels from here down. Efferent. So for sensory, we say PNS to CNS, so peripheral nervous system to central nervous system. And for motor, we say CNS to PNS, so sensory to peripheral. Afferent, efferent. And the integration is them communicating with one another. Your brain or spinal cord determining what to do as a result of that sensory input. Okay, so you're going to see, these are terms that we've brought up previously, but here they are once again. Here's a good example. You see a glass of water, you tell yourself, it goes to your brain, you're like, oh, I really want to drink of that water. So then you send a message to your arm to pick up that glass of water so that you can drink it. Sensory travels up, it integrates, decides what to do, and motor travels down. And it'll be like that regardless. It's never opposite. Okay? Here is a diagram that shows using any generalized receptor. This right here is the spinal cord, not the brain. Okay? This is just showing a rapidness. So receptor, we have an afferent, then we have integration, efferent, and motor. Something I want to point out on this slide, and you're going to see this pattern continue. Everything sensory will always be blue. Everything motor will always be red. So if you're looking at some stuff and it's outlined in red, it's telling you it's motor. And you're going to see that pattern. Same thing with the blue. If it's outlined in blue, it's telling you it's sensory. Okay? Also, and I'll reiterate this as we get further, this structure right here is called the anterior medial fissure. Anterior medial fissure. This is the front of the spinal cord. Sensory information always enters the back. Motor information always leaves the front. So sensory is dorsal, motor is ventral. Always. Sensory is dorsal, motor is ventral. Sensory is dorsal, motor is ventral. I'm a memorizer, so I remembered SDMV. Sensory is dorsal, motor is ventral. When we talk about diseases and damage in a little while, if it's something ventral that's damaged, you know it has to do with motor. If dorsal damage is done, it has to be sensory. Okay? Those are things, these are non-negotiables, that if you can memorize that, it's going to help you navigate through this content. This diagram here divides the two nervous systems. So, nervous system can be broken down into the central, which will be chapter 12, the second one we'll do today, and peripheral, which is chapter 11. The peripheral nervous system has an afferent and efferent division. Afferent is sensory. You can see it's in blue. Look at which direction it's traveling to. The arrow tells you that. Motor is efferent. It's in red. The motor division can further be divided into somatic and autonomic. So now, somatic is our way of saying voluntary. Autonomic is our way of saying involuntary, and they're described there below. And then, of course, it wouldn't be anatomy if we didn't take it a step further and say that our autonomic nervous system, or ANS, can further be divided into sympathetic and parasympathetic. Sympathetic just means it's going to speed up. Parasympathetic means it's going to slow down. Okay? And we're going to go through all of this, but this is an excellent reference slide, and it puts everything in perspective. Again, it's color-coded blue and red, and the arrows tell you if it's going afferent or if it's traveling efferently. Okay? So here you have it for the first time, the acronyms CNS and PNS, the central nervous system being your brain and spinal cord, the peripheral nervous system being your nerves and your periphery. Okay? Your nerves can be divided into two groups, spinal nerves and cranial nerves. Just curious what you think the difference might be. Spinal nerves and cranial nerves. One's attached to the spinal cord and the others are the brain. Yes. Okay? So they're just broken up just based on where they're originating, okay? So you have those 12 cranial nerves and 31 spinal nerves. So you can see here that we are taking just the peripheral nervous system from this point forward, okay? And how I showed you on that big chart that I said this would be a great reference tool, now we're going to take each part and divide it. So starting with the peripheral nervous system, we can divide it into sensory and motor. There's a sensory division and a motor division. Sensory is afferent, motor is efferent, traveling down. For sensory, we have somatic sensory fibers. Just reading that title, somatic sensory. Tell me what you're thinking. Just dissecting the term. Somatic is voluntary. Sensory is afferent or efferent? Afferent. So this is something that I intentionally am trying to sense. Okay, I'm intentionally looking at it, I'm intentionally listening to it, tasting it, smelling it, whatever. Voluntary. Somatic sensory. Visceral sensory. What's visceral? Your organs. So this is sensory information we're getting from your organs. Yeah, like when you're like, oh, my stomach hurts. That's visceral sensory information. Okay? Or I have to pee. That's visceral sensory information. Okay, so it mentions that. Motor. So that motor is going from CNS to PNS. Somatic nervous system. What does somatic mean again? Voluntary. And autonomic means? Involuntary. Right. Okay? So just kind of continuing to subdivide these. Once the vocabulary is going to continue to increase, but if you can catch on to those key terms, there's a lot you can do. There's a lot you can do. All right. So we're on the somatic nervous system right now. The somatic nervous system is voluntary. Tell me what muscle groups have to be involved if it's voluntary. Skeletal. See how you already knew that. You're making connections. This is a domino type effect. It's cascading. So it tells you that here, conducts impulses from the central nervous system to skeletal muscles, because those are what we're voluntarily controlling. This is conscious control of those skeletal muscles. Then we have autonomic nervous system. The autonomic nervous system is us controlling what? Are we controlling it? No. So this is our spinal cord, okay, in our brain if necessary. Controlling it autonomically, which means it's involuntary. There are two options. And the best part about all of this, like we talked about excitatory and inhibitory last week, and we're going to bring that back again today, but there are two options, sympathetic or parasympathetic, and they truly mean excitatory or inhibitory. Sympathetic is you speed stuff up. Parasympathetic is when you slow down. So when you get scared, you're sympathetically dominant, which means everything's speeding up. Your heart rate increases, your breathing rate increases, blood flow increases, that's sympathetic. You're working out, that's sympathetic. I would venture to say that the majority of you are sympathetically dominant. Not that you feel sorry for anybody, but that you tend to be a high-strung type person where you're constantly going. Like, I've got to do this, I've got to get this done. And some of you are parasympathetically dominant, which means that you're kind of go with the flow. I'll just kind of wait it out and see what happens. I guarantee you that if you're married, the person that you're married to is the opposite. Because if you were both sympathetic, it would be bad news. And if you were both parasympathetic, well, nothing would ever happen. You'd just hang out, right? So you have those dominant nervous systems, people who tend to be more relaxed. So if that can kind of put that in your head to kind of give you an idea of what these are like. So parasympathetic is slowing things down. Sympathetic is speeding them up. And we'll talk about a lot of that as we go through. Okay. Neuroglia versus neurons. We talked about this in Chapter 4 when we talked about all the tissues. So the nervous tissue has two types of cells, neuroglia and neurons. I told you the neuroglia are more like the support cells, and the neurons were actually the workhorses. So the neurons actually transmit the electricity. The neuroglia function to support that. What can I do to help you transmit electricity? Okay, so neuroglia are support. The neurons are the actual workhorses. For neuroglia, there are 6 types of neuroglia. And I like this slide, which is why I'm going to stay here for a moment, because it lists all 6 types, and then it indicates to you which nervous system it is found in. So if I'm talking about astrocytes, what nervous system? Central nervous system. And if I'm talking about Schwann cells, the peripheral nervous system. So there's 2 in the peripheral, 4 in the central. Okay? Cool thing about this is that their duties overlap. We just call them something different because of their location. And it gives us more direct guidance if we're talking about them specifically. We know what we're referencing. So the 6 types of neuroglia, 4 to the central, 2 to the peripheral. So we're going to start and kind of describe each one. And I'm just going to highlight, we're not going to go through all the details on each one, but astrocytes. When I think of astrocytes, of course, this is central nervous system, but their job is to guide the growth, guide the migration of young neurons. I feel like these are your parents or your guardians, the ones who are like, this is what you should do, this is where you should go. So these astrocytes bind to them, wrap around them, and kind of show them where they're supposed to go. Astrocytes. Okay, it mentions that it controls the chemical environment around them. So your parents are doing that, making sure that you have, well, if you're at home, not you're the parent doing this, but making sure your kids are taken care of, everything is provided that they need. Okay, so this is astrocytes, support for that. So what we have here, we have this astrocyte cell, and look at it. Right here I have red, and here I have yellow. What do those 2 colors indicate? Nerves and vessels, oxygen-rich vessels. Well, blood can't touch nerves. So what are these astrocytes doing? Here's the nerve that I'm guiding. What are the astrocytes doing to the vessels around it? They've completely surrounded it so that they can't make contact. This is you telling your kids, those are bad people, don't hang out with them. Okay? I'll control this environment, you continue to grow so that we can make sure you're going to be successful. You can see it's anchored to that neuron, but it has surrounded these vessels because blood can't touch nerves. It'll short circuit. This is electricity. We can't have that happening. Okay? We'll talk about here in a little bit what we call that whiteness. Okay? The second type is the microglial cells, or microglial. I call them microglial because I feel like it rolls better. Microglial cells, they basically respond when something goes wrong. So I reference them as being like a little ambulance or a police car, kind of picking up any debris if there's nerve damage done, goes around, cleans it up, puts a little Band-Aid on it, and moves on. Okay? It mentions that these at some point can become phagocytic, which means that if there's trash there, they can pick it up and get rid of it. Okay, so microglial cells. So we have astrocytes, which guide the growth, and we have microglial cells, which respond if there's damage or if something goes wrong. Here's showing you a microglial cell, clearly labeled, and of course you can see neurons here as well. Okay, the Pinnable cells. The Pinnable cells are a little bit more unique because these are actually going to end up forming your triple B, which is what we call the blood-brain barrier. These cells are also ciliated, which means that they have cilia so that they can move things. Okay, they can help move things within that fluid. The fluid that I'm referencing is CSF. CSF is the acronym for Cerebral Spinal Fluid. So it could be 3 words: cerebral spinal fluid, and then sometimes you see it as cerebrospinal fluid. So instead of the old, like cerebral, it's cerebrospinal. Either way, it's the same fluid. Okay, so CSF, cerebrospinal fluid. Because the blood cannot touch the brain, the ependymol cells will create this barrier and will allow nutrients from the blood to diffuse into the cerebrospinal fluid, and we'll get there. So this is withholding any blood from touching the brain, unless of course it's damaged, if there's any type of hemorrhaging. So here are ependymol cells. You can see that they're all joined pretty tightly, and we have cilia here. We have cilia here. Okay, so here would be the nervous tissue. Here's that fluid there. None of that fluid is actually touching the brain. The ependymol cells are protecting it from doing so. Okay, and then we have oligodendrocytes. Oligodendrocytes are an insulation for the central nervous system. Oligodendrocytes function as insulation for the central nervous system. When you have an electrical cord and you plug it into the wall, it's insulated. So that electricity travels straight from the wall to whatever device you have. What happens if that insulation is broken or compromised in any way? What happens to that electricity as it comes through? Does it make it? No, it kind of grounds out, or it shorts out, or if it does make it, it's very slow and only partial. In our central nervous system, if oligodendrocytes are present, it has been what we call myelinated, or it has been insulated. Myelin sheath is synonymous for insulation. It's this insulation that wraps those nerves that help to transmit that electricity. So you're trying to send a message, you want that message to go far. So if I tell my toe to wiggle, it had to go from here to there. It was insulated to get there that fast. You do have non-insulated cells in your brain and in your body, and when they send messages, it literally sparks. So if you've ever watched any type of documentary where it's showing like these little like sparks going on, it's a true thing. Those are non-myelinated neurons. So they aren't insulated, so they just spark like a little firework every single time it transmits electricity. If it's myelinated, then it travels long distances. It's insulated. So myelin sheaths for the central nervous system are called oligodendrocytes. Okay, so far, we have 4 types of neuroglia: astrocytes, microglial cells, ependymol cells, and oligodendrocytes, all central nervous system. Now peripheral nervous system. Satellite cells and Schwann cells. Satellite cells do the exact same thing. Oh wait, here's a picture of oligodendrocytes, sorry. You can see that here's the nervous tissue and it's completely myelinated or surrounded. So it's being protected. And satellite cells and Schwann cells. Satellite cells act like the astrocytes, but for the peripheral nervous system. Satellite cells act like the astrocytes, but they're for the peripheral nervous system. Schwann cells act like the oligodendrocytes, but they're for the peripheral nervous system. So here's how this may play out. Bless you. You're reading a question and it references astrocytes. You know at this point you're dealing with the central nervous system. You're reading a question, bless you, and it references ependymol cells. You know you're dealing with the central nervous system. Those are specific to that. If it says Schwann cells, It's the peripheral nervous system. Remember, and these are only neuroglia, which means all these are doing are supporting the neurons that are actually conducting or carrying out the work. Okay? The difference, there's a small difference in Schwann cells versus oligodendrocytes, and this is also going to be a telltale sign for you as we continue to go through. I'm going to go back to oligodendrocytes. Do you see how this is continuous? There's no gaps. In Schwann cells, there's gaps. Schwann cells are individual cells. And in fact, they're Schwann with a capital S because they're named after the guy who discovered them. Okay, so Theodor Schwann discovered these Schwann cells. And this is an individual cell that wraps around that nerve. You will only find nerves like this in your peripheral nervous system. You'll only find this in your central nervous system. There's no back and forth. Okay? Questions so far? Okay, now to neurons. Neurons, you're going to start off, and it's going to tell us this at the very end of the chapter, you're going to start off with millions and billions of neurons. And a lot of them are going to die by the time they're born, because your neurons are given a goal, what they're supposed to do with their life, and if they don't complete that task, guess what your body does to them? It eliminates it. If you're not going to do what I need you to do, I need to get rid of you. So only a third of those neurons are actually going to survive. They were programmed and told, hey, you're going to control this, you're going to control this. And astrocytes are responsible for guiding them to their spot. Or satellite cells are responsible for guiding them to their spot. But once your neurons are done, and they're mature, they're done. And they become what we call amitotic. Do you recall what amitotic means? They do not reproduce. So if you damage them, it's damaged. Okay? Our skin is actively mitotic. So if we damage our skin, it'll grow back. But our neurons are not. So once the brain is damaged, there's not much you can do for that. Now there's a lot of studies going on, but we're not going to talk about case studies or any of that right now. Every single neuron requires high levels of oxygen and glucose. So your nervous system is responsible for the consumption of the majority of the glucose you take in. In fact, in situations where you're starving, your body will shunt glucose to the rest of your muscles and send it to your brain because your brain needs it. When your glucose levels are low, does anybody know what you do? You usually faint. When blood glucose levels are too low, we go through what we call fainting or syncope, which is like a faint. So you just shut down. That's it. Your kill switch for your brain. Your body's saying, hey, something's wrong. I'm going to tell you right now, bringing it to your attention. You need to eat or increase your glucose levels, or bad things are going to happen. People who are hypoglycemic, this is what we see them doing a lot. Their glucose levels drop too low, they faint. How do we get their glucose levels high again? You just give them sugar. You can give them a candy bar, you can give them Coke, you can give them apple juice, you can give them fruit, anything. They just need glucose. Oxygen as well. The cell body is the area where the nucleus and all the organelles are contained. Here is the textbook diagram for a neuron. So I'm going to discuss this real quick so that you can kind of get the terminology for it. First of all, can you tell me if this would be in the central nervous system or peripheral nervous system? It's for sure peripheral, and we know that because it's got the Schwann cells. Fantastic. Okay? Right here is what we call our cell body. It's also called a perikaryon, and it says this later in your notes. I'm just kind of put it all together while we're here, and a soma. So it's just the control center. Okay? Every cell has it. It's just the shape determines its function. So we have a different layout here. You can see I have endoplasmic reticulum, I have mitochondria, I have all the organelles, just a different layout right here. So here's my cell body. Other things I want to point out, dendrites. These dendrites receive signals. This is the reception point. Dendrites receive. Okay, so there's only two types of signals we can get, and we discussed this last week, but it's excitatory or inhibitory. And remember, your cells immediately sum. If you have more excitatory than inhibitory, we send an excitatory message. If you have more inhibitory than excitatory, we send an inhibitory message. What does inhibitory mean? I'm not doing anything. It inhibits. What does excitatory mean? I'm going to do something. Okay, just keep it simple. If it's excitatory, we're doing something. If it's inhibitory, we're not. Here, right here, is called the axon hillock. This is the part that decides whether it's excitatory or inhibitory. For my brain, I call this the counter. It counts how many excitatory and how many inhibitory. And it decides which way we're going. That is all determined right here. So all this information comes in, and here's where it's summed up. If it's excitatory, electricity is going to go. If it's inhibitory, nothing's going to happen. The axon hillock decides that. So if it's excitatory, that electricity will go. If inhibitory wins, nothing happens. It just hangs out. Here's what we call the axon. The axon is what propagates electricity. So it'll send the message from here onto the next neuron or whatever effector is there. This axon is myelinated by individual Schwann cells. And down here we have the axon terminals. They're also called the terminal buttons or boutons. It's in your notes. I'll point it out when we get there. But this is where we send the information. When we looked at the neuromuscular junction last week, this was the neuro, and then the muscle was right there. So it was an axon terminal communicating to a muscle. So I'm trying to connect everything that you've studied so far, so this isn't so new. Questions on the general anatomy. This right here is what we call a multipolar neuron. And there's only 2 other types, but this is the most common neuron you're going to find in you. Well, we're in human, and never mind. Yes, this is the most common neuron. Okay, so here it kind of goes through each of the parts that I depicted or kind of paid attention to. So it talks about the cell body being the perikaryon, the soma, okay? The term ganglia. Ganglia, you're going to see this a lot. And it's a fancy way of saying that's a bunch of cell bodies together. So you'll hear dorsal root ganglia. Dorsal root ganglia. Dorsal tells me, is it sensory or motor? Sensory. Ganglia tells me it's a bunch of cell bodies. So I have a bunch of sensory cell bodies. Dorsal root ganglia. Okay? And it's usually going to have this little bulge, and I'll point it out when we get there. But ganglia means nothing more than a bunch of cell bodies together. Tracts versus nerves. So this is referencing the axon. The axon is that long part. A group of cell bodies is called ganglia. A group of axons has 2 different names depending on where it's located. If it's in the central nervous system, we call it a tract. T-R-A-C-T, tract. If that group of axons is in the central nervous system. If it's in the peripheral nervous system, we call it a nerve. If it's in the central nervous system, it's called a tract. If it's in the peripheral nervous system, it's called a nerve. A question might say, you're looking at the cerebrospinal tract. Which nervous system will I find it in? Central. These are small words that I'm telling you, vocabulary is over half the battle in this course, and in all of this, for those of you who are going on further and further. Dendrites, this are the input, the sensory aspect of each of these nerves. They receive both excitatory and inhibitory signals. Those signals are what we call graded potentials, and we've discussed graded. Okay, I'm going to come back to that in a little bit, but we have discussed graded. We talked about an action potential, actually doing the work, and graded potential. You were thinking about it, but you didn't do it. But I'm going to come back to that, so don't worry about that right this moment. Here's a good looking picture of a motor neuron. Okay, I already talked about this on the axon, but the axon hillock is responsible for summing. And then the axon terminals or terminal buttons or boutons. This is not a typo. You can say it either way. So this is the very end of the axons that are going to send that message on to the next effector. Which direction do signals travel? They should go from the cell body to the terminal. Like terminal means the end. If that's the case, we call that anaerograde. That's what we want to see, if it goes in that direction. But sometimes you get infections, or you have diseases, degenerative disorders, and they send the message back the other way. The problem with that is what happens to that electricity when it comes back. What happens to that cell body, or that soma? It's damaged when it comes back. Okay, there's risk of damage there. So retrograde is where it's going from the terminals to the cell body. And that's an indication that something's wrong. That's an indication that something's wrong. Okay, it mentions here, if that cell needs to be, we need to get rid of it. If we need to degrade that cell or break it down, it's been damaged. Signal molecules may have that happen. Viruses, bacterial toxins. Bad things are happening. We're destroying those cells. We want it to go anaerograde. We want it to go from cell body to terminal end. The purpose of the myelin sheath is to insulate it. This slide spends a lot of time telling you it just insulates it. If there is no myelin sheath, that neuron is not insulated. If there's a myelin sheath, that neuron appears to be white, so it's what we call white matter. If there is no myelin sheath, it appears to be gray, which is why we call it gray matter. White matter is insulated, gray matter is not insulated. So we would say myelinated neurons are white, non-myelinated neurons are gray. Which one transmits electricity faster, white or gray matter? White. Okay, very well. We know that Schwann cells are found in central or peripheral? Peripheral. And how they work is the individual cell, and this is during development, during your development, okay, or neuronal development, one cell will wrap around that axon, and then it does a constricting action. When I studied this, I thought about a snake. So it wrapped around, and then it continues to get tighter and tighter and tighter, sometimes circling that neuron 50 to 100 times. Okay, but these are individual cells, and they're discontinuous, which means, like in the oligodendrocytes, it was all covered. That would be continuous. These are discontinuous. Discontinuous. So Schwann cells are individual cells. They grab onto that neuron and they continue to constrict in order to better insulate it. Here is a transverse cut of an axon. Here are the concentric circles of that Schwann cell. So you can see that it's wrapped around quite a few times. Okay, that's all that I wanted to show you. White matter versus gray matter. That's what this slide is saying. Is there anyone who's confused on that? If it's white matter in the central nervous system, it's oligodendrocytes. If it's white matter in the peripheral nervous system, it is Schwann cells. Okay? So oligodendrocytes, this is central nervous system. All right. Classification of neurons. Multipolar, which is the one we already looked at. Bipolar, and then unipolar. And this has nothing to do with disorders. Don't cut them. Oh, that's bipolar. Okay, bipolar neurons are usually going to be found in our sensory organs. For example, the retina, olfactory mucosa. Does anybody know what olfaction is? Smell, yes. So olfactory mucosa, that's the lining of your nose going up to your olfactory bulb. So bipolar neurons are not very common. Multipolar are. Unipolar neurons, we're going to find these mostly sensory. So again, it could be special senses, hearing, vestibular, which is balance, and so on. But both of these are not nearly as common as the multipolar. So here's the difference. Here's multipolar, what we looked at. Bipolar, the cell body's in the center. Unipolar, the cell body's off to the side. Okay? when I told you in the muscle chapter that this is a diagram that kind of breaks everything down for you, here's that same diagram, but for the neurons. Okay? And it talks to you about where you can find each of them and where the information comes from. Continuing on to classification, but now based on function. So this was classification based on structure. Here's based on function. Your neurons can either be sensory, motor, or interneurons. If they're sensory neurons, what do? They transmit sensory information from the peripheral nervous system to the central nervous system. If they're motor neurons, they from central nervous system to peripheral nervous system. And if they're interneurons, they're in between, and they're helping to communicate between the two. Okay? So here that tells you what each of them are. And you guys pretty much understand that, so it's not anything really different. Here again you can see that the majority of our motor neurons are multipolar. The majority of neurons we have are multipolar. Okay? So it mentions that there. Examples, but this is using the multipolar, bipolar, and unipolar, and now putting them with their functional classification. So sensory, motor, or interneurons. Membrane potentials. Okay, so we talked about the general anatomy. This takes us back to what we were starting to discuss last week. Okay, we used the term action potential, and I told you that was the all or none setup. And I told you we're using textbook numbers here. So what we're about to look at is this setup. Do you remember what we called this part here? Depolarize or depolarization. And that would make this repolarization. I told you depolarization is not consistent with, but involves what type of ions? Sodium ions. And repolarization involves potassium ions. Okay? We also talked about resting potential, which is where we're getting at right now. And then there will be the threshold, which I'll use a different color. And that resting potential changes depending on what type of neuron that we're dealing with. And I told you once it hits threshold, what happens? You can't turn back. But if you never hit threshold, then nothing happens. Okay? And we're really just going to see a reiteration of that today. So what you're going to see for this chapter carrying on forward is a bunch of redundancy. Okay? So it's going to start off slow and then it's going to become more redundant as we go through it. So it should help you. Okay? It's going to kind of be tight at first and then it should be like, okay, yes, I remember that. I remember that. I remember that. Okay. How do we measure the electricity in our body? What is the unit I told you we were using? Volts. Okay. And technically, we don't use full volts. We're not going to use like volts. Our unit will actually be millivolts. So it will be a lowercase m followed by a capital V. And then we'll talk about our potentials going from negative to positive. At rest, we're negative. Once an action potential is generated, it goes to positive and then it comes right back down to negative. So you're going to see a change in the voltage. Okay? What actually causes that voltage change? The answer is right here. Ions. Okay? You do have electricity going through your body, but how do we change that? How do we transmit it or propagate it? We use ions. Atoms. So there's more positive or more negative. And we'll get there, but I wanted to point that out on this slide. Okay? When we talk about resistance, if a neuron is insulated, it's insulated, so that means the electricity cannot get out. It can only go to its destination. So that's like in an extension cord or a plug. It's insulated, that electricity has a specific destination. It won't go outside of that. So an insulator will have high electrical resistance, meaning it will keep that electricity on track. if it's a conductor, what is a conductor responsible for doing? Continuing on that signal. So it goes from here to here to here to here to here to here. Your neurons range in size. Some of them are a few millimeters. The longest neuron you have is a meter long. Okay, and that goes down to your foot. So they vary in sizes. So sometimes there might be 5 or 10 neurons, or maybe hundreds of neurons to get to a specific destination point. So we need a lot of insulation and a high ability to conduct electricity. Here's a term we referenced. You already know the answers to these. And if you don't, we're going to pull the common sense card. So you have a gate. Okay? And if it's a leakage channel versus a gated channel, what do you think the difference is? If it leaks, something that continuously can get through. If it's totally gated, only certain things can get through. You and I last week used 2 types of gates. Voltage gated channels. So what was required to open and close that gate? Electricity. And we called it, the second one was ligand gated channels. And what was that? Chemicals. That's what we're talking about right here. So a leakage channel is one where there's no gate, things can continuously go in and out. And a gated channel has to be opened or closed. We have chemically gated channels, voltage gated channels. You and I have already covered those 2. The third type is mechanically gated. I'm going to pull a common sense card. What do you think mechanically gated means? Something has to physically move in order for it to open. It's mechanically gated. Okay? So things have to physically move. There has to be a physical change in order for it to open or close. Here are examples of ligand-gated channels or chemically gated. So if the chemical is not attached, then it's closed. If the chemical is attached, it's open. Make sense? What's going to happen when that chemical lets go? It's going to close again. I'm going to substitute the word chemical for neurotransmitter. When the neurotransmitter is attached, it opens. When the neurotransmitter is not attached, it remains closed. Here's voltage gated, and I can tell this is voltage gated because now I have charges, positive and negative. Okay? Here, the voltage is not, it's at rest, so nothing's getting through. Electricity comes through, you can see that the voltage has changed. What was once positive at rest is now negative, so that channel opens and those sodium ions can come in. What happens when that electricity stops? The channel closes. Okay? So gated channels, quick summary, they're gated either based on electricity, chemicals, or physical movement. Okay? And if one of those 3 is not there, then it remains closed, and if one of them is present, then it will open in response to that. Here is where we first introduce our resting potential. These numbers right here are strictly textbook. Of course, I'm going to keep us on textbook throughout the whole course. But if you go into specific patients, you may have different resting potentials, and different muscles have different resting potentials. Or the neurons that control them have different resting potentials, not the muscle itself. Negative 70 millivolts is what we're going to reference as our resting potential. And then it's going to reference here shortly, the threshold will be negative 55 millivolts. So if I want a nerve to do work, I have to give it enough excitation to get it from 70 to 55. So that's 15 millivolts I need to get. What if my resting potential was negative 95 millivolts? It needs a lot more work to get it to do something. It's going to take a lot more to get it to do something. What if my resting potential was negative 60 millivolts? Less work. And I referenced that last week. But muscle or nerves that control your eyes have a really high resting potential, very close to the threshold, because they're constantly moving. It doesn't take a lot to excite them. But your bigger muscles, they have a really low resting potential. So if you want them to work, you're going to have to give them a lot of stimulation. So this resting potential can vary depending on where that muscle is, okay? Or where the neuron is that's controlling that muscle, or effector, okay? So here it mentions that here. What changes this potential? What can take it from negative 70 to negative 55? It's the ions, okay? And here we have a throwback, two throwback acronyms, ICF and ECF. Anybody remember what those may or may not mean? No, let's not go with may not. Intracellular fluid and extracellular fluid. So inside the cell and outside the cell. It should be kind of positive on the outside, negative on the inside at rest. And then we should see it switch. Here we have positive on the outside, negative on the inside. This is my axon. Okay? Sitting here at rest, you can see that I have a voltmeter here. I like to add an extra syllable and call it a voltometer. Okay, it's just because it sounds cooler. But it's measuring the amount of electricity here. It's telling you that this fellow here is at rest. Okay? We know that there are two types of potentials, and we discussed them last week. Graded potentials and action potentials. Graded potentials, what do you end up doing? Nothing. Action potentials, you're doing something. And I referenced you deep couch sitting last week after you were freaking brain fried from this class. And you're watching that SpongeBob and you see that remote when you're telling yourself, oh, but you don't get up to go get it and you just kind of sit back down. Those are graded potentials because you're not actually getting up and doing it. Graded potentials, you're not going to end up doing anything. And action potentials when you said, I've had it, and you get up and go get the remote. Okay? So graded potentials, action potentials. Graded potentials work over a very short distance. Action potentials go all the way. Depolarization, I'm not going to go over this term again, but depolarization is when we go up. Any direction when we're talking about membrane potentials, if it's depolarizing, that means it's going up. If it's repolarizing, it's coming back down to rest, and there's going to be one more term, hyperpolarize. Hyperpolarization means it goes below rest and comes back up. You're hyperpolarized at times when it's fatigued or when it's trying to prevent you from doing something. And I'm going to show you what I'm talking about here in a moment. So depolarization, it says a decrease in membrane potential. And I want to make sure you keep those terms straight in your brain. Because a decrease in membrane potential would be taking it from negative 70 to negative 20. It decreased. If it was an increase in membrane potential, it would take it further negative. Okay? So that's, to me, that was a confusing aspect. Alright? Here, let's say that 50 is my threshold here. Here's just a little graded potential. Because it's depolarizing, is that excitatory or inhibitory? Excitatory. Because it's depolarizing, this is semi-excitatory. Now, we haven't done anything as a result, but this is excitatory. Hyperpolarization, I'm going to increase the membrane potential. I'm going to take it further negative. This is what this looks like. Hyperpolarization. Is this excitatory or inhibitory? Inhibitory is taking you further away. So let's talk about some things that you may or may not engage in. When you drink caffeine, caffeine stimulates depolarization. So it makes it easier for you to be stimulated. When you drink caffeine, you're highly excitable. Some of you. And some of you have built up a tolerance where you have to drink tons of caffeine in order to get that same excitability. Okay? let's say you take some type of depressant, whether it be some type of mood stabilizer, muscle relaxer, something like that. Those stimulate hyperpolarization, which means it takes more work to get you to do something. Okay? A muscle relaxer tells your muscles, you're not moving. Now, can you move? You can, but it takes a lot more work. What does your body want to do? Just lay there. Okay? So when you take some type of stimulant, that is an excitatory graded potential. When you take some type of depressant, that's an inhibitory graded potential. Okay? So this is continuing on graded potentials, saying that they only travel for short distances. And in order for us to generate an action potential, we have to sum up all the graded potentials. So if I get 50 excitatory potentials and 10 inhibitory, what am I doing? I'm doing something. I'm excitatory. Can you remember the part of the neuron that counts all that up? Axon hillock. Yes, the axon hillock sums all that up. Do you remember this guy? Good, because we're not going to discuss him. Okay, here it talks about the threshold being at negative 55. It's saying negative 50. These are textbook numbers. Okay, they can change. Usually the peak of an action potential is somewhere around positive 30 millivolts, and it's just temporary. It goes straight back down. It tells you here that for depolarization, we have sodium going in. Did you already know that? Yeah, you did. Okay, and it tells you for repolarization, there's no sodium going in, but right now I have potassium going out. Okay. Hyperpolarization, there's no sodium going in, but there continues to be a little bit more potassium exiting, which is causing it to go a little bit more negative, but just temporary. So it mentions here, this is natural, this is not drug-induced or anything like that, but there's a slight dip below the resting potential, so it's slightly good, but then it comes right back up. That's your body's way of telling it to not do things over and over again, which would lead it into tetany. This is a healthy cycle. So you're here at rest. Whatever type of stimulant you have, it sums up and you hit threshold, so it generates an action potential. So we have depolarization, repolarization. As it's coming down, it kind of hyperpolarizes and it goes right back to rest. It's just temporary. And you can see here, we're looking at time in milliseconds. Like we haven't even covered a full second yet, how fast that this is happening. Like when you study this process, you're like, oh my gosh, how is it that I'm even holding my pencil right now? Because this happens in split seconds, literally. Okay. Questions here, or what we've covered so far? Okay, so the role of the sodium-potassium pump. And this was brought up last week. Sodium-potassium pump is responsible for changing the membrane potentials, so that positive and negative. Okay, you're not going to have to know any more than that. But the sodium-potassium pump is what's changing the intracellular and extracellular fluid and those ion charges. We know that threshold is the point that has to be reached in order for an action potential to be generated. Does everybody understand that? If threshold is not reached, what happens? Nothing. Nothing happens if threshold is not reached. The all or none principle says once the threshold is released, once threshold is reached, an action potential will be generated. There's nothing you can do. I mentioned it and referenced it to pulling the trigger on a gun last week. Once you pull the trigger, you can't catch the bullet. Like, oh, I didn't mean to do it. I mean, you could, but it's not going to end up well for you. Okay. So here it shows you, and it has it color-coded right here, so you can kind of have an idea. So we're going from resting in yellow to action potential in red, and then hyperpolarization in gray. So a natural progression should be from yellow, red, gray. Yellow, red, gray. And it doesn't do the whole thing at once. This is your brain telling it to do something, and it travels down. It doesn't just light up all at once. It travels down. It's what we call propagation. So here you can see, and the potentials are changing, positive and negative charges. Okay? Any questions on that? I didn't ask. Okay, absolute versus relative refractory period. I mentioned this last week as well. Absolute refractory period is where no matter what, it will not do more work. It doesn't matter what stimulant is there, it will not do more work. Absolutely no more work during this time frame. And again, this is a split second. Relative refractory says, I'm on my way to relaxing, but if you entice me enough, I may do something for you. Okay? So relative versus absolute refractory. So we have absolute refractory. Here's our resting. No threshold is really depicted here, but we have depolarization. While it's repolarizing, this is absolute refractory. Here, I'm on my way down to rest. And now I'm in my relative refractory period. I want to get to rest, but if you stimulate me enough, I'll go and generate a second action potential. During absolute refractory, no action potential can be generated. Relative refractory, an action potential could be generated, but it's going to take a lot of stimulation. Okay, because we're going down, hyperpolarizing, and then back to rest. Go ahead.
I'm sure you said it, but
Go ahead.
Depolarization, if it doesn't get past threshold, is that still depolarization?
If it does not hit threshold, it could still be, depolarization just means it's moving up.
Okay.
An action potential is when it hits threshold and goes up. So, continues on its way up. So anytime it slightly comes up, it could still be considered depolarization. But that could just be a graded potential.
Okay.
Once it hits threshold, it immediately becomes an action potential.
Now, does it always get hyperpolarized whenever it goes down?
It should, if it runs its natural cycle. It should be depolarized, repolarized, hyper, rest. And even if you read an ECG or EKG, how it's that's what's happening. Depolarization, hyper. Like all of it. Now you look at that stuff and you know what it means. You're not just gonna be that bystander. Like, what does that mean? You know. You could be the doctor. Don't be the doctor, but you could be the doctor. All right, conduction velocity. How fast can we transmit electricity? Well, it depends. It depends. If I'm completely myelinated, real quick, in the central nervous system, if I'm completely myelinated, I can transmit electricity pretty quickly. If I'm not completely myelinated, I'm going to transmit it a little bit slower, but I can still transmit it super fast, and we'll continue with that in just a moment. Here it brings up some conditions to bring up or to mention. Multiple sclerosis. Multiple sclerosis is where you start to build these plaques on the myelination, and it actually begins to harden that insulation, and as a result, the electricity is not transmitted, and so you begin to lose control of the muscles that those nerves innervate or those nerves touch. Multiple sclerosis usually happens a little bit earlier in life in comparison to most conditions. It's a little earlier in its onset. We do have treatment for it. It is becoming a little bit more common. I have to say that for sure. You hear about people developing it more or beginning to show symptoms of it. There are treatments for it to help you kind of slow the process down, but it doesn't cure. There's no cure. Okay, you can just kind of slow it. Nerve fiber classification. This continues with the speed of the conduction of velocity. Velocity of conduction. Diameter, degree of myelination, and speed. We just discussed degree, but I'm going to show you what I'm talking about here. Group A fibers. If I have nerves that have very large groups, or a very large diameter versus a very small diameter, which one's going to transmit electricity faster? The bigger one. So the larger the diameter, the more area I have to transmit electricity, the faster I can transmit electricity. So large diameter, my group A fibers, they can transmit electricity at around 150 meters per second. You're not even bigger than 3 meters, and you can transmit electricity at 150 meters per second. Like that. Even faster than that. Okay? Group B fibers, these are intermediate, and they can still transmit electricity quickly, 15 meters per second. That's 5 times your height. I mean, that's giving you 3 meters. You're not even 3 meters. Anyway, group C group C fibers are much smaller in diameter, and it mentions here that they're unmyelinated. So the fact that they're not myelinated means that their conduction velocity is going to be slower. But nonetheless, 1 meter per second. That's still pretty quick, but not in comparison to 150 meters per second. So these that have this really large diameter are myelinated, highly myelinated, transmit electricity very quickly. Intermediate diameter, lightly myelinated. Small diameter, non-myelinated or unmyelinated. This right here talks about the neuromuscular junction. And I'm not going through all the steps again. For me and for you. Okay, but do you recall what the synapse is? So there was the axon terminal, and then there was that moat that they called the synaptic cleft. I call it a moat. And then it had the actual motor end plate. So the synapse is where those two come together. Okay? Did they ever physically touch? No, there was a gap. So that's what we call the synapse. I'm going to ask you again to use a little bit of common sense, but we're going to hear the term presynaptic and postsynaptic. Where do you think presynaptic is? Before. Postsynaptic? After. So here's the synapse. If it's before it, it's presynaptic. If it's after it, it's postsynaptic. Okay? So we're going to use those terms, presynaptic, postsynaptic. Presynaptic is bringing in that action potential. Postsynaptic is the one receiving it. It mentions here, I'm going to draw this for you. It's not going to be as cool as it's drawn or depicted in your book. And this is clearly not a full neuron. I get it. But let's say you have this neuron. Here's the synapse, and here's this neuron. And it continues. So this is presynaptic, and this is postsynaptic when I'm referencing this synapse. But when I'm referencing this one, this is now presynaptic. This is postsynaptic. Why would it continue to move in any direction, whether it's afferent or efferent? Why would I continue to see movement? Why would it not just stay here and this always be pre and this always be post? What's the electricity going to do? It's moving. So it's going to presynaptic here, it's going to send the message here. This is going to receive it. It's postsynaptic. Then this one needs to send it to this one. This one needs to send it to this one. So when you talk about the synapse in presynaptic and postsynaptic, it mentions that most neurons function as both. At one point they'll be presynaptic, at another point they'll be postsynaptic. Does that make sense? Okay, so here, this is just showing you, like in this case, that's presynaptic. You can see the axon terminals attaching to the cell body here, there would be dendrites there. This would be postsynaptic. Then when we're talking about this one, presynaptic, post, and so on. Well, actually, those are terminal ends there. But this would actually be the postsynaptic for all of those. I should not have tried to explain that without looking at it first. Synaptic cleft, I've already touched on it. That's that moat. What travels across the synaptic cleft? Acetylcholine, which is a classification of neurotransmitter. Acetylcholine is an excitatory neurotransmitter. So here it references the term chemicals. Chemicals are going to be some type of neurotransmitter that's going across that synaptic cleft. What happens to the neurotransmitter once it's done? It could either be broken down by an enzyme, it could diffuse away, or it could be resorbed by that neuron, that terminal end again. Okay? This is nothing different than what we discussed last week. It's just putting it in, reintegrating it into this chapter. There is a gap. So there is a delay. But is it a really long delay? No. I didn't mean to say it like that. Like, there's not a long delay. You can tell yourself to wiggle your toe right now and wiggle your toe. There wasn't much of a delay. It was still a delay, but it wasn't much of a delay. Okay? Postsynaptic potentials. They can either be excitatory or inhibitory. Get ready for this. EPSPs, IPSPs. A fancy way of saying excitatory postsynaptic potential and inhibitory postsynaptic potential. Why? Why do we have to make it so difficult? Yeah, because you're doctors. Okay? You're doctors. So they're like, oh, well, we're receiving EPSPs. That just means that you're getting excitatory information. Okay. IPSPs, that's inhibitory postsynaptic potentials. It's telling you you're not going to do anything. But regardless if it's excitatory or inhibitory, what part of the neuron is going to determine what you do? The axon hillock is going to add it all up. And if excitatory trumps, you're going to do something. If inhibitory trumps, you're doing nothing. Is everybody okay with that? This is where I'm telling you it becomes redundant. We're going to add a few terms in, but it's redundant. Okay? Bottom line, you can either be excited or inhibited. Okay? So here it tells you that regardless of the EPSPs or IPSPs that you receive, the axon hillock will determine what you do. It will sum them up. So what I just said just a moment ago is here in writing on this slide. Here, was this excitatory or inhibitory?
Which one is it on?
Right here. Is this excitatory or inhibitory? It's excitatory because it's depolarizing. Did it reach threshold? No. So this is what we would call a graded potential. This is what we would call a graded potential. It was a slight depolarization, but it didn't reach threshold, so nothing happened. This is an excitatory postsynaptic potential. Inhibitory. I gave you the answer. This one right here is doing what? It's inhibiting. What's that term for going less negative, more negative? Hyperpolar. It's temporarily hyperpolarizing it. Is this graded or action? If it doesn't pass threshold, it is not action. Graded. Doesn't pass threshold, graded. You are trying, and that's we all ought to be doing this. There's some of you are just using your faces, and I get your faces. I'm cool with that. Okay. All right. Once more, this slide says it's going to sum the EPSPs and the IPSPs, and it's going to determine whether it should be excitatory or inhibitory. Okay? There are different classes of neurotransmitters. Neurotransmitters are the chemicals that we use to tell us to do something. We're just generally referencing them here. First of all, the one that we should be familiar with is acetylcholine. Acetylcholine was the one that was released at the Neuromuscular Junction, which is a junction between a neuron and a muscle. I'm going to go ahead and give you. I knew you knew that. I know you knew it. Whatever. Anyway, acetylcholine is excitatory. So it tells that muscle to contract, to do something. Yeah. Contracting is moving. I'm good with that. And when we're done with acetylcholine, what's going to break it down? Acetylcholinesterase. You should just say that at Thanksgiving or something, just so your family's like, gosh, you're so smart. I know. Other types of neurotransmitters. Biogenic amines. This right here tells me, and I did add a syllable, I normally do that on things that I don't think sound natural, but this tells me these are protein-based because it looks like amino. The fact that it's protein-based means it can't get into my cells. This is like your epinephrine, your norepinephrine, your nitrous oxide, epinephrine, norepinephrine. We're going to talk about nitrous oxide on the gaseous one, so let me take that. Serotonin, your feel-good, and histamine present whenever you're having a response to something, an inflammatory reaction. Could be allergic, it could just be that you had damaged tissue or something like that. But none of these can get into your cells. Here, gas and lipid are gasotransmitters. We have nitrous oxide. If you stay with me for 2402, nitrous oxide is one very excitatory, and it increases blood flow crazy fast. Nitrous oxide is involved in learning and the formation of new memories, which means when you're excited, it's easy for you to remember things. Okay, it's also, and I keep saying nitrous, but it's nitric oxide, sorry. And nitric oxide is also the main neurotransmitter necessary for the blood flow to increase to the penis for a man to maintain and achieve an erection, or achieve and maintain an erection. So nitric oxide comes into play all over. This is a really important neurotransmitter. But if you can be stimulated while you're learning something, you'll remember it, which is what we're going to talk about in Chapter 12. Endocannabinoids. These are similar to THC, which is not okay, because we're talking about anatomy and we're helping people and we're not involving ourselves in this, but ones that are similar in their structural component and actually act chemically the same are believed to be involved in learning and creating memories. It may also be involved in neuronal development, which means our neurons developing, controlling our appetite, and suppressing nausea, which is to an extent why people say that they're used for medicinal purposes, but we're in Texas. So here it talks about the neuronal integration, neuron integration. 4 groups of neuron integration. Simple neuronal pools, we'll talk about discharge zones, facilitated. Let me just show you a picture. I'm going to cover all of this, but I want to show you a picture. Here, we have presynaptic, and these would all be postsynaptic. Here is what our main discharge zone. Here is a facilitated zone. So that's what it's talking about. This one is right in line with that. These ones are off to the side, more peripheral. But still, I have presynaptic, and these would all be postsynaptic neurons. So we're about to talk about the 4 classifications of neuron pools. Diverging, converging, reverberating, and parallel. Can you have any guess as to what diverging means? It's going away. What does converging mean? All coming together. Okay, reverberating, any idea there? Something into reverberation, it's kind of just doing the same thing over and over again. Okay, and then parallel after discharge, I'll show you that. So, you might have a neuronal circuit set up like this. This is what we call a diverging circuit. What this may be is my brain telling me, hey, I need you to get up and do something. So this neuron sends a message, and it has to tell my legs, my abdominal muscles, my arm, everything to work so that I can stand up. Okay, so that would be a discharge circuit. One neuron sends a message, and it activates a lot. Discharge, or divergent, sorry. Convergent. Convergent is where a lot of different stimulatory sensory information has to come in order to get you to do something. My easiest reference to this is when you walk into your grandma's house, you know what you're going to see, you know what you're going to smell, you know what you're going to hear, you know how you're going to feel when you're there. Okay? Now, let's say you walk into a store in the mall and you smell the smell that smells like your grandma's house. It's going to bring that vision of your grandma's house, all of that back in. So you have a bunch of sensory information all coming together to give you that output. Okay? This is a lot in the recollection of memories and the creation of memories. You're taking visual, auditory, olfactory information, emotional information, and creating memories. Okay? So a lot of input to one direct output. So converging, coming together. Reverberating, this is doing the same thing over and over again. So this would be some type of task like walking. Thank goodness for you. Some of you may have to remind yourself to take steps or watch where you're walking because sometimes you don't pay attention. But a lot of times you can start walking and you'll continue to walk even though you do other activities like talk or hold your kid or whatever it is that you're doing. Because your brain is doing a reverberating circuit. It's saying, pull that muscle up and you continue to walk. Okay? When you're ready to stop, you have to send a message from your brain to tell your muscles to do what? Stop. Okay? So something that's repetitive. Parallel after discharge. This is one of those situations, and this is the difference between the basic high school student and a successful college student. Your ability to think critically. So here you have a problem. Now you need to pull in all of these different resource materials. Once you pull all of these in and kind of put all of that data together, then you write down your answer. So this gives you an example of a math problem, but this would be like a case study. Like you have a patient, and the patient comes in and they have a rash. Okay, so a lot of patients come in with rashes. So based on your experience, what other exposure have you had to this? What other symptoms do you have? Is there swelling? Is there redness? Is there pain? You pull all of this in, and then you make your diagnosis. So this is a critical thinking type thing, where you're pulling a lot of information in. You have that problem, and here's your output, but you're bringing in a lot of other things in order to figure out what your output should be. This right here is the difference in successful college students and practitioners as well. So if you can think critically, you're going to be pretty successful. It's not necessarily do you know the material, it's can you think. That's a true statement though. Anyways, developmental. When you're a baby, your nervous system comes from your ectoderm, which that's a biological concept. We are triploblasts. This is the outer layer that forms our nervous system. Neuroblast, and neuro tells us it's nerve, but what does blast tell us? They're mature. They're done. They become amitotic. Wait, totally messed that up. Neuroblasts are the baby ones. Sorry, in my head I heard myself say it, and then I was like, what? These are the baby ones, but they become amitotic, which means they do not regenerate. So they'll migrate to where they're supposed to go. What guides them to their destination? The A1. Astrocytes. If it's in the central nervous system and if it's in the peripheral, it's a satellite cell. Growth cone. This is what I was telling you at the beginning, that the majority of your neurons, so this is a colored one. This isn't exactly what it looks like in you, obviously, because that would be pretty cool. But anyway, you have millions of these nerves that shoot up as you're developing, and they're told, you go touch this muscle. If they don't make it, they're destroyed. They have to, by the time they're finished developing, touch something. And if they don't touch something to control it, they're destroyed. So we call those growth cones, because it started to grow and then it's trying to stretch out and touch something. Again, a lot of those are going to die before you're even finished developing. If they don't do what they're supposed to do, they're gone. Okay, let's take a break, and then we'll come back for chapter